program of brain-derived neural stem cells. It's kind of a general title, but more specifically, what I was looking at was variations in gene expression programs between different mouse strains. So, an overview of my presentation. First, I'll be giving some background information on neural stem cells, epigenetics, and MECP2, which is our gene and protein of interest. We'll move on to discuss the relevance to child health, and then some preliminary observations, which provides the basis for my project. I'll show my results and draw some brief conclusions for suggesting some future directions. Okay, so first, neural stem cells. Neural stem cells, which I'll abbreviate NSCs, are the progenitors of neurons and glial cells which comprise the central nervous system. Early in gestation, NSCs will divide symmetrically to increase their own population before later on dividing asymmetrically, producing first neurons and then later neurons, astrocytes, and oligodendrocytes. The process of going from an NSC to one of these differentiated cells is known as differentiation. And though it's not completely characterized, it is understood that in addition to things like transcription factors and cell-to-cell signaling, epigenetic mechanisms will play a major role in NSC differentiation, especially in the determination of cell type-specific expression programs. So what is epigenetics? Epigenetics is defined as heritable variations in gene expression that don't rely on changes to the underlying DNA sequence. The major epigenetic mechanisms are DNA methylation, histone modifications, activity of non-coding RNAs, and chromatin remodeling. And these will act to either turn genes on or off or increase or decrease the activity of certain genes. Today I'll be focusing on DNA methylation. DNA methylation in mammals occurs on the cytosine residues of CpG dinucleotides. The main proteins involved in this process are the DNA methyltransferases, which will first deposit a methyl group to cytosine, and then the TET proteins, which will catalyze a further modification to hydroxymethylcytosine. 5-methylcytosine, I'll abbreviate 5-MC, and 5-hydroxymethylcytosine, I'll abbreviate 5-HMC. Just to underscore the importance of this modification, roughly 70 to 80% of CpG dinucleotides are methylated in the human genome. And the effects of DNA methylation on transcription and chromatin are mediated in part by methyl binding domain or MBD proteins. The most abundant of which is methyl CpG binding protein 2 or MECP2, which is our gene and protein of interest, which will bind to both methylated and hydroxymethylated DNA. MECP2 has a wide array of functions, including transcriptional activation and transcriptional repression, with the recruitment of protein partners, as well as chromatin compaction and chromatin loop formation. MECP2 is widely expressed in various tissues in the body, though the highest expression is in the brain, lung, and spleen. Focusing on brain, the expression will be highest in the neurons, with lower levels detected in the glia, which leads to one of the main physiological roles of MECP2 is in neuronal maturation. And we'll see that the expression of MECP2 will actually increase as neurons mature. So this brings me to why MECP2 is relevant to child health, and that is abnormalities in MECP2 expression and function are implicated in several neurodevelopmental disorders, including Rett syndrome, autism, fetal alcohol spectrum disorders, and MECP2 duplication syndrome. Just to focus on Rett syndrome for a moment, I'll be coming back to it later. Rett syndrome is caused by mutations in MECP2, generally a loss of normal function. It affects 1 in 10 to 15,000 young girls, making it the leading cause of mental retardation in females. These are quite prevalent disorders. The molecular mechanisms of these disorders are still not fully understood. What is known, though, about MECP2 is that MECP2 expression and function is critical in neurons. Here on the left, I have a schematic of a normal neuron, with here at the bottom, I have the dendrites, the cell body, and the axon. You can see that with altered MECP2 levels, being an autism neuron, we see a reduced cell body size as well as reduced axonal branching. Moving to the MECP2 mutation, which is Rett syndrome, in addition to the reduced cell body size and reduced axonal branching, we see reduced dendritic branching as well. And here on the right, I've shown, on the left, we see Rett syndrome neurons, which have these characteristic features, but when MECP2 is added back, you can see a return of larger cell bodies and increased branching of the axon head. MECP2 is a relatively well-studied protein, though not much is known about its role in neural stem cell differentiation. So here we have the in vitro model we're using for neural stem cell differentiation. Embryonic NSCs are isolated from the E14.54 brain and cultured. When these cells are cultured in vitro, they will aggregate together to form these balls of undifferentiated self-renewing cells called neurospheres. Take this as time point day 0. These neurospheres will be dissociated to single cells and cultured to form these differentiated progenies. The indicated time points here of day 1 and day 8 will represent early and late events in neural stem cell differentiation respectively. This system has been used before to characterize the expression programs of genes important to neural development, as well as to design MECP2 gene therapy strategies. There are several models for Rett syndrome, having mouse models having different mutations in the MECP2 gene. Here indicated on this graph, we have at normal 100% MECP2 function, we'll have the normal phenotype, whereas the MECP2 deletion over here will have the most severe phenotype. What's interesting here is that based on the different mutations, we see a wide range of different phenotypes. And not only do we see that in a loss of function and more severe phenotype, but also in a gain of function or overexpression, we'll see that a more severe phenotype. Basically what I'm trying to show here is that varying levels of functional MECP2 will have an effect in changing this phenotype. Two mouse strains that are commonly used for MECP2 research are the CD1 and BL6 mice strains. The main difference between these two is that the CD1 is an outbred mouse, which means that we'll have a greater degree of genetic diversity, making it better for things like toxicological studies, whereas the BL6 mouse is inbred, therefore having a reduced genetic diversity, making it better for genetic manipulation studies. And in fact, the majority of the Rett syndrome mouse models are the BL6 mice. These strains have reported differences in behavior and physiology for things like anxiety, emotion, stress response, as well as neuroanatomy and survival of transplanted neural stem cells. And both these have been used before in the Rastegar lab. Which leads me to my preliminary observation, which provided the basis for our project, is that when we look at the undifferentiated self-renewing neurospheres, looking at the CD1 mouse, at the protein level, MECP2 is widely expressed throughout the cells. However, when that is compared to the BL6 mice, we see here that only a few cells are expressing it. So there's quite a large difference between these two strains, which led to the hypothesis that the expression programs for MECP2 and related genes during neural stem cell differentiation must be strain specific. So the plan for my project was to determine the expression levels of MECP2 and DNA methylation related genes at the transcript level using RNA samples from the embryonic forebrain and differentiating NSCs from both the mouse strains to compare them and see if I saw any more differences. The MECP2 related genes talking about include MECP2 E1 and E2, which are the two protein isoforms, the two isoforms of MECP2, which share common functional domains, and BDNF, which is brain-derived neurotrophic factor, which is the most studied downstream MECP2 target, which plays a role in neuronal development, maintenance of synapses, and that kind of thing. I also looked at DNA methylation-related genes because MECP2 is a binder of methylated DNA, and the expression of MECP2 often correlates with the global levels of DNA methylation. So I looked at the DNA methyltransferases 1, 3A, and 3B, which are responsible for the deposition of the methyl group, as well as the TET genes 1, 2, and 3, which are responsible for the conversion of 5-MCs to 5-H's. So the aforementioned RNA samples were converted to cDNA and analyzed using quantitative real-time PCR. The experiments were performed in triplicate using 3 independently obtained RNA samples, and then were analyzed for statistical significance using the Relevance software. So moving on to my experimental results here. Just to explain the graphs here, the black bars will represent the CD1, red bars representing black 6. At the bottom I have the indicated time points in either embryonic or differentiating neural stem cells. So first I looked at MECP2, E1, and E2, and noticed that in fact, just like the protein expression, we see a statistically significant difference in the day 0 undifferentiated self-renewing neurospheres for both the E1 and the E2 isoforms. In addition to this in-strength difference in expression at this one time point, we also see for the E1 isoform a different expression program where from day 0 to day 8, the expression of the E1 isoform will decrease, whereas in the black 6 mice, the expression from day 0 to day 8 increases, so that at day 8 we have roughly the same value. So after looking at this, I thought, well, there must be an effect on the downstream target genes of MECP2. So we looked at B and F, the brain-derived neurotrophic factor, and determined that indeed, like MECP2, the expression of this gene is much higher in CD1 at day 0, which indicates that this effect seen in MECP2 is not isolated to the one gene, but in fact will have a knock-on effect to the other target genes. After looking at this, I moved on to the DNA methylation related genes, expecting to see some differences. However, we determined that there was no statistically significant difference in the expression of DNMT1 and 3A at any of these time points. I also looked at DNMT3B, but did not detect any amplification in this system. So after looking at the DNMTs, I moved on to the TET genes, where I saw something interesting here. I have the graph separated. I have day 1 neurospheres on the left, and day 8 relate differentiation events on the right. At day 0, I saw, much like BNF and MECP2, that the CD1 mouse has a higher expression of all 3 of the TET genes. However, at day 8, this trend switches, and we see that in black 6, the expression of these genes is higher. It'd be interesting to look at this data with the global hydroxymethylation data and see if they correlate. So some brief conclusions from this. I've determined that there are statistically significant interstrain variations of the expression programs of MECP2 and DNA methylation related genes. The important message from this is that it is important to take this variability into account when establishing controls as well as comparing experimental results. Some possible future directions for the study would be to look into more downstream MECP2 target genes, especially those important in neural development, to determine the extent of the variation between the 2 mouse strains, as well as to determine the global methylation levels during NSC differentiation of those strains, determine how these correlate to the DNA methylation genes. I actually tried to start looking at this, but I didn't get any reliable results. So now I just have to thank Dr. Rastigar first and foremost for the opportunity to work in our lab, as well as all the Rastigar lab members, Vici, Robbie, Harl's down here, and Sean. And thanks to Mitch for the opportunity.
Thank you very much, Ern. Very interesting. Are there any questions from the audience?
Maybe just quickly, because we're out of time here. So you said that B6 were more anxious in terms of a mouse strain?
It varies on the studies.
Okay. Is there has anyone done any sort of cognitive tests on, like, maze or obstacle or, catch and receive, and then, sort of different you know, tests like that or between these two mice strains to see if they're even at this sort of basal level differences without actually inducing any mutations, because these are wild type mice, quote unquote. If there's any, you know, differences that can be pulled out in terms of behavior, quote unquote, intelligence or response timing or stress or anything like that.
There have been some that I've read, but I can't think of any of the specific results right now. But I have read studies where they had different stressors, forcing the mice to swim and stuff like this, to see their different responses.